Okay, class, this is the second part of this lecture about bioelectricity. So we're going to go over electromyograms now to finish off this lecture. So by now we should have covered um, where bioelectricity comes from, what are the origins in terms of like the cells, and we should have even gone over some ECG, electrocardiogram type of material. So electromyograms are essentially the same thing as electrocardiograms. The biggest difference is that there are biomedical signals or electrical signals that are coming from muscles that are not necessarily from the heart. So this could be muscles in your arm, in your legs, anywhere where there are excitable muscles. You can measure an electromyogram. So let's move on to the next thing here. All right. So where do they originate? So generically, muscle cells are called myocytes. There are three types of muscle cells. There's cardiac, which we covered with the ECGs. There are skeletal and smooth muscles. What's important for us are skeletal muscles. Smooth muscle cells are the muscles that are lining some organs like your kidneys and your liver. Skeletal muscles are the ones that make up tissue that are connected to your bones. So those are the ones that cause locomotion or movement because they're connected to your bones, and when they're excited, they can contract and make your body move. So that'll be the topic of this portion of the lecture. All right, I'm not going to go into too much detail about the actual muscle cells, but what's important is that they are long, they're cylindrical, and they're striated, and they basically make up. So here's an actual muscle cell here. They're made up of small fibers called myofibril, and they have multiple nuclei, and their cell membrane is made up of sarcomella. Their cell cytoplasm is called sarcoplasm. And these myofibrils are made up of even smaller filaments, which are called myofilaments. And these are all parallel to each other. What's important is that you have like kind of this hierarchy of you have a muscle cell, you have micro fibrils, and also microfilaments or myofilaments. Sorry. And so at the myofilament level, you'll see that you have some small molecules that allow you to have muscle contract to cause movement. And so I don't think we get into that in the next slide, but just know that I guess the important thing here is that these muscle cells are all innervated, so they have some neurons that are connected to these excitable tissues. And this is going into detail again. You have myofilaments, which are the small fibers that are within the myofibrils. There are two types of myofilaments. There are thin ones. They're the ones that contain actin and troponin, which are the ones you probably have heard of from high school physiology. And then they have thick filaments, which are made up of myosin. And basically they work together to cause sliding between those structures, which cause contraction of the muscle. I don't want you to really memorize any of this with the exception of kind of the hierarchy where you have a muscle fiber, you have myofibril, and then myofilaments within the myofibrils. And that's basically it. What's important is in the next slide here when we talk about how do you excite these things. So now that you've kind of seen generally what a muscle cell looks like, at least, maybe not necessarily how it functions, but there is some contraction that goes on with these muscle cells, and so that contraction comes from the fact that you have these nerves that are going towards the muscle, and so we call those neurons motor neurons, and they originate from the spine. If you remember from our previous lecture on the spine and the central nervous system, and so you have these motor neurons which then synapse onto the muscle fibers. So remember that synapsing means that you have the axon, which then they terminate to these processes which create little areas between the axon, the neuron, and the neck and the muscle fiber, or the muscle in this case. And within those junctions, you have the releasing of neurotransmitters which cause sodium to move in and out of the cell, which then cause contraction. All right, so how does this work? So the entire process here from motor neuron to muscle, that whole system is called the neuromuscular junction. And so the way this works is that you have an action potential, which again we should have covered where that originates, moving along this neuron and going through each of the processes here. Once the voltage changes at these individual synapses, that causes some release of neurotransmitters, and those neurotransmitters then act up on the ion channels within the muscle fiber. That opens up sodium, which then causes a membrane potential to change across the membrane of the muscle cell. And that also causes action potential if it's a big enough stimulus, which then causes the contraction of the muscle. Yes, so in total, this neuromuscular junction functions as a motor unit. So again, this neuron originates from the spinal cord, which then causes this contraction. The next slide. All right, so this is just detailing what happens at the synapse if you wanted to know the biological processes here. So as the nerve impulse comes down one of the nerves, that creates a membrane potential across the neuron, which causes these small neurotransmitters to be released. Those neurotransmitters then attach to the ion channels of the muscle cells, and that again causes an influx of sodium to basically go into these ion channels, which then cause a change in membrane voltage on that muscle cell. That's basically how this works, and specifically for this, it also uses some calcium to release some of that neurotransmitter. Again, the details are not important here, just the general process of how this works, going from an electrical impulse, which is a membrane voltage that's changing along the length of this neuron, to a chemical release back to an electrical impulse. That's kind of what's important in terms of the take-home message here. Okay, so with all that said, what do you actually measure? And we're not measuring cell by cell, right? So there's essentially lots of these neuromuscular junctions that we can measure. So there's millions of them, and so when we get electrodes, we'll talk about electrodes later on, and technically how they have to function and how they have to attach to your body. Once you have those, then you can start measuring some signals. So remember that you have millions of these neurons and these neuromuscular junctions in any part of your body. So the summation of all of those action potentials within the muscle and correspondingly the actual contractions can be measured on the surface of your skin. Now they're very small voltage signals, and they can be compounded by noise, but we'll get to in ECE 503, if you guys enroll in that, how to actually get rid of that noise and get these relatively clean signals. So muscle contraction is due to change in relative sliding of the myofilaments, and that's induced by these electrical impulses. And so that's the electromyogram. So what each of these are showing the summation of all of the neurons that are firing off to essentially make the muscle contract in a specific location. This might be, this is an amplitude of that voltage, which is, sub millivolts over time. You can start seeing some patterns if you have a specific kind of disease. So this is a neuropathy. So, if there's some neurons that are dying in that innervation, or if some of the muscle is dying, you can get some characteristic readings from these electromyograms, also called EMGs. So I don't know if we go over in this lecture how to actually place the electrodes, but I think in the next lecture we'll go over that. So here's kind of a kind of a small look into that. So let's say you have a muscle. Obviously, you don't have a muscle exposed like this. You would have skin, like shown here. And what you would do is you would place some electrodes, and you have to place them strategically. And remember that muscle is innervated by motor neurons originating from the spine. So like if you were to get a magical electrode that you can actually see what the potentials were along the neuron, you would get these nice clean signals for each of the different nerves originating from the spine to the muscle. But in reality, that would be very invasive, right? So these are individual action potentials along the actual length of the neuron. We can't actually measure that unless we actually poke that specific nerve in close proximity. So what we're going to have to do is, to be non-invasive, we have to have electrodes on the skin, and what you'll read is this kind of noisy looking signals, and these noisy signals are the summation of all of the individual motor action potentials that you're measuring from each of these. Now imagine, this is not to scale, right? Here you only have 1, 2, 3, 4, 5 individual action potential waveforms. You can have millions of these, and they all sum up to give you the raw signal that you're measuring. So if you're interested in a specific disease or application for measuring EMGs, you'd have to understand something about the muscle that you're studying, and also what nerves are innervating it, so that when you get the raw EMG signal, you're able to decompose it into individual action potentials. And so this is, again, just showing you that what you're measuring on the skin is a summation of different action potentials along each neuron. All right. Now this is where I guess I'll talk about how to place electrodes. So the most important thing is that you place the electrodes near the muscles of interest. And specifically, you want to place the electrodes in the direction of the predominant fiber direction. Why is that important? Because we want to measure the voltage signal along the length of the muscle fiber. So if we place the electrodes near that fiber direction, then we're able to see how the signal is traveling along that direction specifically. The other important thing is that you want to keep one of the electrodes grounded, and to do that you'd have to place it in a spot in the body where there is no tissue that's excitable, and what that means is that there's no tissue that is producing an extra electromyogram, basically. So you place that kind of near a bone or near some spot where there isn't any nerves that can be giving off a signal. Remember that the ground or the reference is supposed to be a fixed voltage from which all other voltages or other electrode voltages are measured between. So that's important as well. So there is a little bit of an art to placing the electrodes and making sure that you get some nice clean waveforms. Here's an example of that. You can't really see it, but this is from a paper where they are measuring the EMG on the thighs, and you can't see the electrode placements, but they do place some electrodes along the length of the muscle on the legs, and then they place the reference electrodes near some bone or some junction where there is no neurons firing off. Okay. All right. So that's as deep as I'll go into electromyograms. On the same note, there are electroencephalograms called EEGs. Now these are the potentials that are produced by the brain, and let's get into that here. So just like with the muscle, where you have hundreds of thousands, if not millions, of nerves innervating a muscle cell, or muscle cells, I should say, there's an estimated 10 to the 11th number of neurons in the brain. Specifically, a lot of them are on the outer layer of the cerebrum, which are called cortical neurons, and they're all strongly interconnected. So for instance, a single neuron will approximately, this is a wide range obviously, but will approximately connect to 1,000 to 100,000 synapses. That's quite a number of connections that one neuron can make. And if you have 10 to the 11 neurons, you can see what kind of connections you'll get. Um, because of this, you're able to measure some relatively strong signals at the surface of the scalp. And you can do so using electrodes, just like with the electromyogram. What's shown here is kind of a visual of what you might see when you're measuring an EEG. So you have multiple electrodes that are sitting on the scalp of a patient. Now, you might have seen this and you might have thought like, oh, this is like sci-fi kind of stuff, but this is actually how you measure an EEG signal. We'll get to the standard leads here. This is showing you the kinds of the relative amplitudes. Actually, there should be numbers here. I'm sorry. This can be on the order of a couple of millivolts to microvolts in amplitude. And the frequency, it surprisingly has a range of frequencies up until 50 hertz, which is relatively large bandwidth considering that a lot of the processes in our body are not high frequency. Okay, so where do you place these things? You need quite a bit of electrodes to be able to measure EEGs. So the standard EEG placement is called the 10-20 system. So 21 electrodes are placed on the surface of the scalp. There are two of those 21. There are two that are reference electrodes. One called the Nasion, which is right above the bridge of your nose. That's near the bone on top of the skull here, in front of the skull. And then you have the Indian, which is right behind the bone, the rear bone of your skull. Again, you want to place the references where there's minimal innervation, so that would be a great spot to put these in this case. And then the skull is divided in transverse and median planes, and they basically divided in 5. So you have the 10%, 10%, and then they're divided into 20% increments along the perimeter of the skull in the transverse and median planes. These placements are standardized by the American Electroencephalographic Society. So you have to place these electrodes relatively equidistant from each other. And then there's additional placement of electrodes. Like one, for instance, called the Queen Square system. That's just another place to put electrodes. So these are just standard electrode placements. You can then add on to these if you want to do like bipolar or unipolar electrode measurements. And just for your reference, like these, the naming conventions are listed down here. So like if you see an electrode that's called F, that has to do with the frontal part, the frontal cortex. You have the O, which is the occipital lobe. So you're able to measure, temporal, central, occipital, parietal, frontal kind of signals along the brain from those. And so obviously there's more electrodes here than there is up here. These are just additional electrodes you can place along the head, for instance. Okay, you can have bipolar versus unipolar. All that means is that in the unipolar fashion, the electrode potential from one electrode is in reference to a neutral electrode. So that would be like the Nasion or the Indian. Or they can be in reference to the average of all the other electrodes. In bipolar, you would be measuring the electrode potential between two specific electrodes. So this is kind of most like the ECG that you saw during the actual lecture. Okay, so here are some basic kind of characteristics of an EEG wave. You can basically break them down to alpha, beta, delta, and theta waves. They all have different frequency ranges, and they're all present at different states of awakeness or consciousness in a person. So for instance, beta waves are higher frequency, so they're from like 13 to 30 hertz. They're usually measured towards the front of the brain. You have alpha waves, which are from 8 to 13 hertz. They're usually measured in the occipital lobe, well, again, having to do with eye movement. The theta waves are much lower frequency, like 4 to 8 hertz, and they're usually measured in children or when you're asleep. You have delta waves, which are very low frequency, on the order of 0.5 to 4 hertz, and this is seen in sleeping adults and in babies. And then you have random spikes that can be due to other diseases or like epilepsy, for instance. And these also show you relative amplitudes. So, okay, so what's the difference here? So it turns out that when a person is asleep, the EEG that you will find it actually depends on the state of sleep, but they tend towards lower frequency and higher amplitude. So they tend to be delta or theta waves when you're sleeping. When you are awake and you're actually, and you're cognitively functioning, you tend to have EEGs that are higher frequency and have lower amplitude signals. So they'll be more like beta waves and alpha waves in that case. Okay, so I mentioned to you that during sleep it does depend on your sleeping state, and that's because when you are asleep, you go through different phases of sleep. You've probably heard of this. So when you're awake, you have these rapid changes in the EEG. When you're in light sleep, you still have rapid changes for the most part. When you're in REM sleep, REM is rapid eye movement sleep. That's when you're dreaming. Your eyes are moving as well while you have your eyelids closed. That's also very rapid. So it's kind of like you're having beta and alpha waves. But then when you're in deep sleep and you're not dreaming, you see that the EEG turns into something more low or high amplitude, low frequency. So again, that's when you are talking about like delta waves and even some theta waves. And then obviously, if you don't measure anything, that's just cerebral death. All that means is that you're brain dead, and there's no activity originating. That's because no neurons are firing off for a given time. So you can do all sorts of things. You can even do these kind of EEGs for different cognitive tasks. And why is this important for you guys? Because at some point, if you're going to go into this field, you have to be able to measure these signals and then decompose them down and essentially figure out for a specific signal what task is the person engaged in. And if you're, say, trying to make some kind of brain-computer interface, you have to be able to translate these EEG signals into something that the person is actually doing. So it is important to understand where these signals come from and the kinds of amplitudes and frequencies that they produce. So here's an example of where you can take the neuromuscular junction that you learned about in muscles and even EEGs and essentially make a device that helps people with neuromuscular diseases. So like I've named a couple here where you have cerebral palsy, multiple sclerosis, and ALS. These have different diseases. Like for instance, ALS, the motor neuron that innervates the muscle is dying, and there's a deterioration in that motor neuron. So with these people, they lose a lot of their movement function because their motor neurons are not working. So we can bypass that by basically interfacing with the central nervous system and then stimulating with some stimulators to regain some function. This is a very hot field of research. There's a group in this class doing their paper on this, but I encourage you guys to look at this specific paper. So go to this link and read that paper and also look at the video, and you can see that this is how you marry basically muscle stimulation with EEG measurement and combine those together to make a system that can actually help people with these diseases. Take a look at that, and I think you'll enjoy it. Okay, so the next class, we'll get to specifics about bioinstrumentation. So what kinds of circuits do we need to measure these signals like ECG, EEG, and EMGs? What are these electrodes? What are they made out of? Are they just pieces of metal, or are they special types of devices? You'll see that. And a little bit more about bioelectronics in that class as well. Okay, thank you for listening, and that is all. I'll